we're going to get started with our next chapter. And the nervous system is actually split into several sections. And this first part of the nervous system is on the basic structure and function of different parts of the nervous system. So we're going to talk about different types of nerve cells and the functions of those, and also the functions of different parts of the nervous system. We'll talk about things like neurotransmitters and some other structures as well. All right, so again, just an overview of the nervous system, starting with what we know the nervous system is. It's going to be the system that is pretty much connected to all other systems, definitely connected to the muscular system. We even have nerves that innervate bones. We have nerves that are connected to glands, and they communicate with even the integumentary system to sense things that are on the outside. So it's connected to a lot of systems so that we can maintain homeostasis. Within the nervous system, we have two types of cells, and those two types are the neuron and the neuroglia. And I like to think of these two as the neuron being the Beyonce and the neuroglia as being her entourage. The neuron is going to be the main player. It's going to be the one that is going to do everything that we know the nervous system does. It's going to send and receive nerve impulses or send and receive signals. So it's the main player, the star. So that's the Beyonce. And then we also have neuroglia. Neuroglia is the entourage. So it's there to make sure that the star is getting any support, also making sure that it's getting the nourishment that it needs, so it can help support the sending and receiving of the signals. And so that's what the neuroglia is there for. Again, that's the Beyonce is the neuron, is what the main player, the star player of the nervous system. And the neuroglia will be the cells that are there for support, for nourishment, to make sure that Beyonce is fed and so that she can do her job. And so if you look at this picture here, the neuron is this large star here, or this large cell. And then we have neuroglia that are just kind of surrounding this cell, and this is going to be the support system for the cell, the neuron. Also, we can divide the nervous system into two divisions, and the two divisions are the central nervous system and the peripheral nervous system. So if you were to take the nervous system and kind of make a flow chart of it, you'll have one that's the CNS and the other that's the PNS. And the CNS is going to be our brain and spinal cord. And then our PNS is going to be all the nerves that come from the brain, and all the nerves that go towards the brain. And so we have cranial and spinal nerves that kind of stem out into other parts of the body. It's there to connect the CNS to everything else. Okay? And we can also kind of subdivide those divisions a little bit farther. And I'm going to take this chart and actually kind of turn it around. So I'm going to go back to where I had this here. And so the PNS can actually be subdivided a little bit farther into two. If you look at this chart, this is the CNS, this is the PNS. The PNS is actually divided into two portions, the sensory and motor. So back to my chart, we have motor here. We have sensory. So those are the two divisions of the PNS. And then going back to this flow chart, the sensory division is going to be what's taking in information. We'll talk about this a little bit more. And then the motor division can actually be subdivided even farther into the somatic nervous system and the autonomic nervous system. So I'm going to go back to my chart. The motor can be subdivided into somatic and autonomic. Okay. And somatic is going to be, if you want to maybe make a note under this will be voluntary movement. And so skeletal muscles fall under here. So this is voluntary movement. Whereas autonomic or automatic, the autonomic nervous system is those things that are involuntary. And so we think about involuntary motor functions. Those are things like the heart, many of our digestive organs, like our stomach and our intestines, the uterus, the bladder, all those things are going to fall under the autonomic nervous system. And so if you notice here, we have the motor division subdivided into somatic and autonomic, and it's smooth muscles, that's the ones I just named. We also have our cardiac muscle, that's the heart, and then glands actually go under here as well. So this chart is the same thing as this one. I've just kind of changed the flow of it, and in my opinion, it's better to understand in this way. If you prefer the other one, that's perfectly fine. But just know that we have several divisions of the nervous system itself, the CNS and PNS. This is the central nervous system, which is the brain and spinal cord. We also have the PNS, which are the nerves that stem from the brain and the spinal cord. And then we have from the PNS, those are the nerves that stem from the brain and spinal cord, carry information to the CNS, and those that carry information away from the CNS. These are a motor division. And so the motor division can then be subdivided into two, the autonomic and somatic nervous system. And then we may go into a little bit more detail with this one within the nervous system. But the autonomic nervous system can actually be subdivided a little bit farther. I'm going to add this to it into what we call the sympathetic and parasympathetic nervous systems. And the sympathetic nervous system is going to be fight or flight. This is what's going to speed up the heart rate. And then parasympathetic is going to be more of what we call rest and digest, where, for instance, while you are possibly listening to this video, your heart rate is lower, unless of course maybe my voice scares you, which I hope not. But your heart rate should be low. Your breathing rate should be slow. Your food is digesting. Anything that you've may have drank is going through your bloodstream and then being processed by your kidneys. So all of this is happening through your parasympathetic. Sympathetic nervous system is when you're fearful, anxious, nervous. For instance, if you have some type of attacker, your body is going to increase your heart rate so that you can pump as much blood to your arms and legs so that you can either flight, run away, or fight off your attacker. And then you'll have increased breathing so that you can have as much oxygen going to those limbs as possible. And some other functions will be slowed down while those things are going to speed up. So again, these are the divisions of the nervous system. We'll kind of work through each of those. And this is the same chart that's here, just a different way to look at it. This is just showing that sensory receptors, for instance, maybe sensory receptors on the skin, the eyes, that sensory divisions of the peripheral nervous system will take that information to the CNS. The CNS will then bring that information back out, and it will then connect to the motor division because it's taking information back out to either a muscle or a gland. If it is a skeletal muscle, that's part of the somatic nervous system. And if it's any type of smooth muscle, cardiac muscle, or gland, as part of the autonomic nervous system. So, three general functions of the nervous system. Just like I mentioned, we have sensory, motor, and then you have to have something that's going to process information. So let's go first with sensory. Sensory is just taking that information in. That's why it says input. So the input is what's being taken into the CNS. So it's going to be responsible for receiving information. This could be coming from the eyes, it could be coming from the skin, it could be coming from the mouth, whatever, to receive information that something is happening or coming into the body, or to help detect any changes. So again, carried to the CNS. So for instance, let's say we have touched a hot stove. If we touch a hot stove, we have sensory receptors on our hands that's going to sense it, and it's going to take that information to the brain or spinal cord. Then we have to have something that's going to process that information. That's the integration function or integrative function, where it's trying to process, trying to make a decision. This is the CNS making a decision about what has been brought to it, about that sensory information that has been brought to it. Once it makes a decision, so we touch a hot stove, that's sensory. It processes that information saying, you just touched a hot stove. We need to do something about this. So what are we going to do? We're going to send a signal back to the muscles within your arm. This is going to be the output. The decision has been made and acted upon. And so it's going to push that back out to those muscles in your arm so that you can pull your hand back. And whatever it sends a signal to is called an effector. An effector can be a muscle or a gland. The example that I was just giving you, for instance, if we are touching a hot stove and we pull our hand back, those muscles contract so that we can, and so those muscles are the effectors. Another example would be we go outside, realize that we have sensory receptors that are receiving the stimulus of it being cold. It takes the information to the CNS. The CNS will then process that information and make a decision about what needs to happen. And then because we're cold, it may take that information back to a muscle to help us contract. It may take that message to a gland, specifically the sweat glands, to inactivate them. Or if it's hot outside, again, take that information, it's processed, then take that information to an effector, specifically the sweat gland, so that it can activate them in sweat so that we can cool off. And so the motor division, again, is taking information from the CNS to the PNS, and it's going to be carried to specifically effectors. And as I mentioned before on our other flow chart that we just wrote, there are two divisions of the PNS, which are the somatic and autonomic nervous system. The somatic nervous system is going to be voluntary, so those things that we can control, whereas the autonomic nervous system is automatic, and so those are things that we cannot control. That's going to be like heart, any other smooth muscles and glands, like we mentioned on the other slides. So now we're going to talk about, again, that main player, the Beyonce, and the different parts to that specific type of cell. If you remember, we already know what a cell looks like. We know parts of the cell, and we know, we have a nucleus, and we have a cell membrane, and all of that. And we also know that certain cells are very specialized. For instance, the muscle cell is very specialized for a muscle. It's going to have a ton of mitochondria. It's going to have the sarcolemma. It's going to have the sarcoplasmic reticulum. So everything is very specialized for that cell, and it looks a little bit different from the normal cell that we see. And so a neuron, which is a nerve cell, also is going to look a bit different from the regular cells that we know of as being a cell or an animal cell. So we have a couple of structures. All of them will have a cell body, also called a soma. This is just this center portion right here. Within the cell body, we have a nucleus. Everyone knows what the nucleus is. It's like the control center of the cell that is where it's going to house DNA and all the above. We're also going to have a ton of organelles in this area, and just other just substances that help to make the cell, keep the cell's structure. We also have dendrites. Dendrites are what's going to, one of the projections that stem off of the cell body, and they're going to be receiving information. They're going to be responsible for receiving information and bringing it into the cell. And then we also have axons. The axon is going to be taking information in and pushing it out towards maybe another axon, or not another axon, another neuron, or maybe even taking the information and sending it to a gland or a muscle cell or any of the others. Okay. Also, just kind of talking about a little bit more, we have within the cell body neurofilaments, which are very similar to some of the filaments that we refer to when you talk about just regular cell structure. Is there for support, for structure, to give it its shape, and also support. And you find that within this area. And then the chromatophilic substances is very similar to the ER, where it's going to be responsible for making proteins or making whatever else it needs within the cell. Okay, and then we have our dendrites. I just talked about the dendrites being the receivers of information that takes information towards the cell body. And it could be getting information from anywhere. It could be getting information from a muscle, a gland, it could be sensory information, whatever, but it's receiving information and taking it to the cell body. And then we have the axon. The axon is going to be responsible for taking information away from the cell body and taking it to maybe another cell, a gland, or a muscle. We have a couple different parts. The axon hillock is simply this area where it kind of starts to branch from the cell body, just this area right here. Collaterals is just the areas where they start to branch off. Axon terminals, terminal means the end of something. So it's just the end of the axon. And the very, very tip of the axon, we have the synaptic knob. And the synaptic knob is going to be where neurotransmitters are going to come out of, or going to be transported to, so that it can act on something else. And so one of the types of neuroglia that we mention, or that we will mention, is a Schwann cell. And if you all remember, I just said that the neuroglia is going to be responsible for support. It's going to help to allow the neuron to do what it needs to do when it comes to transmitting signals and information the way it needs to, so that action potentials can happen. And so the Schwann cell is one of the first ones, and it is going to be responsible for producing myelin. And myelin is made up of lipids and proteins. It's what we call a lipoprotein. But it's going to have this cell. This is the nucleus of that cell. It produces this lipoprotein, and it creates this sheath. And the sheath is just these layers of material that wraps around the axon. It's kind of like an insulation. You know, if you have a cord, your phone cord, computer charger, there's insulation on the outside of that cord, or the covering to that cord. That's what the Schwann cell produces. That's what the myelin sheath is. And it helps to transmit the information. And this is, the same analogy is if, the insulation starts to come off of your cord. sometimes you may get a bit of a shortage in your cord because the insulation helps to transmit that information. And so we'll kind of talk about how if we don't have this insulation on the axon, how that could affect the transmission of information as well. All right, again, just a little bit more. Our Schwann cell, again, the Schwann cell is going to be one of the neuroglia that's going to be responsible for secreting myelin, which is that lipoprotein. And it starts to coat it in such a way that it makes what we call a sheath. This is just layers of myelin that's been produced. And that coating creates this insulation around the axon. And so this is going to be the axon, and we have this myelin sheath that coats it. But if you notice, we're going to go back to this picture. We have myelin sheath here, and there's a bit of a space. Myelin sheath, a bit of a space. You go a little bit closer, myelin sheath, space where you can actually see the axon. Myelin sheath, space. Myelin sheath, right? So those spaces between each one, that's the myelin sheath, Schwann cell that makes the myelin sheath, there's a bit of a space in between the two, and that's called the node of Ranvier. The node of Ranvier, we'll talk about a little bit more detail, but because it has those gaps in between each sheath, that helps for the transmission to actually go even faster. And we'll talk about what that's called later on. So, we talked about how this myelin is very similar to a cord that has insulation around it. And so it's very important for these axons to be myelinated. And those that are myelinated, those that need to be, they are going to be able to transmit impulses extremely, extremely fast. And wherever you find axons that are myelinated, you're going to find what we call white matter. And I'm not sure if any of y'all have ever dissected a brain before, or if you've ever seen a picture of the brain that's been cut. You'll see that we have what we call gray matter and white matter. The white matter of the brain is white because it has the axons in that area, and those axons are myelinated. And the gray of the brain, or the spinal cord, is gray because it has what is not myelinated of the neuron, that's the cell body. And so the cell body is going to be in the areas that we call gray matter. Also, those that are unmyelinated, they don't have the Schwann cell, they don't have that coating around it, so they may not be able to conduct the impulses as fast. An example of a disorder where the myelin sheath is not properly coating an axon, or maybe where the myelin sheath has pretty much been destroyed, is multiple sclerosis, or MS. And some of y'all may have heard of it. You may know of someone that has it. You may even have it. But it is a result of the myelin sheath being broken down. It's actually an autoimmune disorder where the body actually produces antibodies against the myelin because it doesn't recognize that the myelin is self. And so it starts to attack it and break those myelin sheaths down. And so those people will have issues when it comes to conducting the impulse the way that it needs to. And so they may not be able to have a proper muscle control, atrophy may happen, weakness, a lot of different things when it comes to what needs to be done, or what nerve impulses help the body to do. So, we talked about neurons as far as the basic structure, the general structure of them, but they don't always have the same type of structure. They don't always look like the ones that we've been looking at, which are these. But most of the ones that we have in the body are going to have that particular shape. And these are called multipolar neurons. And these three examples are actually named based on the amount of projections or the amount of processes that come from the cell body. And so, multi, you all already know, multi means many. And so, we have many processes from the cell body. You notice we have several processes that are stemming from the cell body. So, this is a multipolar neuron, and most of the ones that we have in the body itself, most of the ones in the CNS are going to be multipolar neurons. We also have bipolar neurons. Bi means two. So that means we have from the cell body itself, we have two projections from the cell body. And so that's how we can identify a bipolar neuron. And then we have uni. Uni means one. So from the cell body itself, don't get confused because you see two things going in this direction, but from the cell body itself, there's only a single process or projection. And so these are going to be very limited in the body. These are going to be found in sensory places. And failed to mention that this is specifically in those that are those special sense organs. So like the eyes, the ears, the nose, and things like that. So these are special sense organs. You'll find bipolar neurons. As far as sensory neurons, you'll find our unipolar neurons. So if you remember when we talked about the functions of the general functions of the nervous system, 3 words: sensory, motor, integration. You'll see those 3 things a lot. And if you just get that down pat, a lot of this you're not really relearning or learning anything new. You're just kind of taking the same information and applying it in a different way. So then we were talking about the functions of the nervous system. Here, we're using those same 3 things, but we're talking about classification of neurons. So we have neurons that are responsible for sensory. So a neuron that's taking in information to the CNS, just like the functions. Okay. We already talked about how most of them are going to be unipolar from this slide. Most of them are unipolar, but we do have some sensory that are going to be our special sense organs. So they're bipolar neurons. After it gets that information, it's going to take it to the central nervous system for integration. Remember, that's another general function of the nervous system. These type of neurons are called interneurons, very similar to integration. This is going to be responsible for association, processing, making the decision. They're going to be linking and connecting the sensory neuron to the motor neuron. The motor neuron, we're going to have mostly our multipolar neurons, and they're going to be responsible for taking information away from the CNS into a muscle or a gland. Another thing I failed to remember just on this slide, another name for sensory neurons is afferent neurons. So if you see afferent or sensory, they both mean the same thing. And motor neurons, another word for those, efferent. So sensory or afferent, interneurons or association or integration. And then we have motor, the efferent neurons. So same exact concept as what we talked about a few slides ago. This time it's simply talking about classification of neurons in general. And we also can classify neuroglia and talk about neuroglia a little bit, as where they're located and what they do, but they have a wide range of functions. They're going to help with making those synapses, the connection between the neuron and something else. It's going to help give it nourishment. It's going to help provide structure. And we'll talk about all of these in the next few slides. Just a wide range of functions for neuroglia. And again, this is the entourage. This helps make sure that the neuron itself is able to do its job. And so some of the neuroglia that we have in the CNS, I want to emphasize that the ones that we're talking about now are found in the CNS. So these are in the brain and spinal cord only. So the very first one are astrocytes. Astrocytes are called astrocytes, astro because it's a star shape. Astrocytes are going to be responsible for making sure that it gets its nourishment. Okay, so it's going to be connecting it to blood vessels. So it makes sure that it gets nutrients, it makes sure that it gets the oxygen, everything that needs to happen to feed the neuron. That's what the astrocyte does. And it says also as part of this blood-brain barrier, which is just the barrier that makes sure that what's inside the blood that's not supposed to get inside the brain is not able to, or is prevented from getting to the brain, and vice versa. Our next is our oligodendrocytes. Oligodendrocytes are going to be responsible for, again, in the CNS, for producing myelin. And I'm hoping you're thinking at this point, oh, we've already talked about a type of cell that produces myelin. And we have. We've talked about the Schwann cell that produces myelin. But the Schwann cell is not found in the CNS, okay? So the oligodendrocytes, they do the same thing that the Schwann cells do, but they only do it in the CNS, whereas the Schwann cell does it in the PNS. The next, we have microglia. Phagocytosis. Maybe we remember phagocytosis is where we are engulfing, is a type of endocytosis of solid particles. So it could be engulfing something that is foreign, that should not be there. It could be engulfing dead or decaying cells, brain cells that need to be engulfed because we don't want just dead cells hanging around. It could be engulfing any type of foreign particles or bacteria. That's what the microglia are there for. And then we have epidermal cells. Epidermal cells are going to be responsible for producing this barrier around the CNS, the brain, and the spinal cord. It helps to also regulate cerebral spinal fluid, which we'll talk about a little bit later. And kind of cover the brain and spinal cord. Just an extra barrier. This picture here just kind of shows all the ones of the CNS. We have our oligodendrocytes that do the same thing as the Schwann cells. This is just in the CNS. We have our astrocytes that are connecting the neuron to blood vessels. We also have epidermal cells that produce this extra layer, this barrier, so like the bodyguards. And then we have microglia that are there to engulf any solid particles that should not be in the area. And so now we're talking about specifically neuroglia in the PNS, okay? And so these two are only found in the PNS. We've mentioned Schwann cells several times already, producing myelin so that we can speed up the nerve impulses. The next is the satellite cells, and satellite cells are going to be responsible for supporting bundles of neuron cell bodies. So sometimes we have several neurons that kind of work together, and they have to be bundled a little bit closer so they can do what they need to do. And that area where they're bundled is called ganglia. You'll talk about this a little bit more in detail, but ganglia, again, is just a bundle of neural cell bodies or neuron cell bodies. So, I know many of y'all have heard of if a nerve cell dies, a lot of times you won't regain the function back. Not saying that it will never happen, but nerve cells, if damaged, if the actual cell body is damaged, which is what this one is talking about, is usually going to die. Okay? And the thing is, after our neurons have matured, we don't have more neurons that are created. For instance, with skin cells, we scratch off skin cells, they're constantly being regenerated. We injure ourselves, they're constantly being regenerated. It does not happen like that with nerve cells. And regeneration is going to be a lot different in the CNS and in the PNS. So the first one is talking about regeneration in the PNS. And remember, we have our cell body, and we have our dendrites, definitely again not the best drawer, and we have our axon. And over the axon, we have our myelin sheath, right? And there's going to be a better picture, of course, on the next slide, but just to kind of see what I'm talking about here. If this axon of the peripheral nervous system is injured, you have an accident, and what is injured is the axon, not the cell body, but the axon. Usually, this, like if it's injured right here, this part that's downstream will kind of degenerate. Okay? The axon itself. But the myelin, the Schwann cell that produces the myelin, it will stay there. And so since it will stay there, it can now guide the growth of a new axon. And so this part here that's upstream, this axon that's here, can start to regrow. And there's a better picture on the next slide. And then if it regrows in such a way that those axons are going to connect and the transmission of nerve impulses can happen, then that nerve will be back to normal. And that's, you've seen people that may have lost the function of a limb and then eventually start to walk again. And for instance, my father, he had an accident where he was unable to use his arms for quite a while. And after a while, maybe a month or 2 or 3 months, he slowly but surely was able to regain the control of his arms because those axons were able to reestablish a connection. Now, the regeneration of the CNS, it's going to be a bit different. So the axons don't have neurulema, the axons, they don't have neurulema in the CNS. So we don't really have that guide that we need like we had in the PNS. And the Schwann cell could possibly proliferate, make more, but oligodendrocytes, they don't. So after they're injured, they won't make any more. And so more than likely, if brain cells or cells within the spinal cord, if they are damaged, more than likely the cell will die and regeneration is not going to happen and function may be lost. This picture here kind of shows the regeneration of the PNS. Again, this is the injury of the axon. Everything downstream will start to degenerate, but it could start to continue to grow. And if those connections are formed, then the neuron will have its function, its original function, or be able to function the way it used to. So, synapse. Synapse is a word that you may have heard or you may have recognized. Synapse is simply the connection between two things, and one of those things will be a neuron. It could be a neuron and another neuron. It could be a neuron, a gland, or a neuron and a muscle. In this case, we're talking about two neurons. And so we have the synapse or the connection. And again, they don't necessarily touch because it still has that synaptic cleft that's there. But the two neurons get very, very close. And we have what we call, if we have two neurons that are synapsing, the presynaptic, pre means before. So before the synapse is going to be the one that's sending an impulse. And so if this is a presynaptic neuron, it's going to be sending an impulse to this neuron. Okay? And the postsynaptic neuron is what's receiving that impulse. So in this case, this will be the postsynaptic neuron. Okay? So presynaptic is going to be sending the impulse. Post, after the synapse. Pre is before the synapse. Post is after the synapse. So the neuron that's after the synapse is going to be responsible for receiving that information. And so, whenever information is brought to a neuron, we're going to talk about a little bit more detail, but it travels down the axon. This is what's taking information away from the cell body and down towards the axon. Whenever this signal or this impulse reaches it, the synaptic knob, which remember is the very end of the axon terminals, when that impulse reaches here, we will then have an influx of calcium ions. At that point, when calcium starts to enter into the cell, that's going to trigger the neurotransmitters to be released from those vesicles. Once those neurotransmitters are released, they will then bind to receptors on the postsynaptic neuron so that it can exert some type of effect. It could be one to excite the postsynaptic neuron to make it do something, or it could be something that will stop it from doing something. Maybe it's to inactivate a gland. If we are cold, we want the sweat glands to be inactivated. So it could be something to inactivate or inhibit a neuron. So now we're going to talk about how nerve impulses are actually sent and what happens as nerve impulses are sent. So, the cell membrane of a neuron is usually charged. And when we say charged, think back to chemistry, the basic chemistry. That means it has either a positive charge or it's negatively charged. Okay, it has a slight charge to it. And when something is charged, it is polar. In this case, we call it polarized. Y'all remember, even in chemistry, some biology, how you have a polar covalent molecule, which means that it's slightly positive or slightly negative on certain ends. And so this neuron, the cell membrane, is usually charged. And very important, the inside is negatively charged and the outside is more positively charged. So this was our cell. We have more negativity on the inside than we do on the outside. As far as the distribution of ions, because it's not distributed evenly, we have different ions or amount of ions on the inside and the outside. So, what ions play a big role? Sodium and potassium. Okay? So, again, we have our neuron. As this says, potassium are the major intracellular. Intra means inside. So we have mostly potassium ions on the inside. And we also have sodium ions, but they're the major extracellular. So they're mostly on the outside. So we have mostly sodium on the outside. And the way that we make sure that they are the way that they're supposed to be, whether the cell is at rest or whether nerve impulses are being sent, we have a pump, the sodium potassium pump, that helps to regulate where they need to go. We also have other ions, and this is what's going to really make the cell membrane polarized or charged, or, like we said, more negative on the inside, is that we have other ion channels that are going to allow negative charges on the inside of the cell. I think this is going to come up also on the next slide. Maybe not. But we have more also, in addition to sodium and potassium, we have specific ions that can go in and out. And so overall, it's more negative, again, on the inside of the cell in respect to the outside of the cell. Okay? So, when a cell is at rest, that means that there's no signal being sent, it's just at rest. Again, we still have more potassium on the inside of the cell. More sodium. More sodium on the outside of the cell, but this area here is still more negative. The inside of the cell is more negative, and the difference between the two, we call that a potential difference. Specifically, it's a negative 70 difference, or 70 is going to be the difference between the two, which means that the inside has a negative 70, or a charge of a negative 70 compared to the outside. So at rest, the inside of the cell is a lot more negative, specifically negative 70 millivolts, than the outside of the cell. Okay? We want this to remain the same if there's no nerve impulse. But even if there's a nerve impulse that's sent, this is going to all change. But then the sodium potassium pump will regulate this back so that it can get back to the way that it was, where we have a resting membrane potential at a negative 70. So remember, resting membrane potential, negative 70. If there is nothing that's being conducted after the nerve impulse is being been sent, we always reset back to a negative 70. So, we know that neurons are not going to always be at rest. They're going to be sending and receiving signals all the time, depending on the stimulus that it receives. So, that means it can become either more negative or less negative. Okay? So, again, remember, we have all these ion channels. Go back to this picture here. All these ion channels that are allowing potassium and sodium in and out, and then other ions in and out. And depending on the ions that go in and out, that's going to make the cell membrane potential either more negative or less negative. And remember, the resting membrane potential, I'm going to write this off to the side just as a reminder. The resting membrane potential is equal to a negative 70 millivolts. So if this membrane potential becomes more negative, we call that hyperpolarized. If it's more negative, that means that it has to be less than -70. If the cell membrane potential becomes less negative, that means becoming more positive, we call that depolarized. So this is where it's greater than -70 millivolts. Okay? And that can change back and forth again, depending on ion channels that we have. But the way to start an actual action potential or nerve impulse, we have to reach a threshold. So this is the minimum amount that we need to cause an action potential to happen. And so the threshold potential, which means what is the minimum amount that we have to get to in order for an action potential to happen, is -55. So, not a lot of math, but I want to ask, if we are trying to reach an action potential and we need to get to a -55, our resting membrane potential is a -70. So do we need to become more positive or more negative? Think about that. So if our resting membrane potential is -70, in order to reach an action potential, we have to reach the threshold of -55. So are we becoming more positive or are we becoming more negative? Hopefully, this simple math has told you that we're becoming more positive or less negative, right? And so if we're becoming more more positive or less negative, that means that we have to depolarize. Okay? We're moving closer to zero. We're trying to not be as polar, because remember, polar means charge, right? So we're depolarizing until it reaches, again, this threshold potential. And at that point, we can now have an action potential, signals can be sent. So let's look at this chart. Okay? Resting membrane potential, again, is 70. I'll write this up here again. Resting membrane potential, -70. All right? So we start here, resting membrane potential. If you notice, this is -80, -60, -40, 0, +20, and 40, right? So we start at -70 at rest. If you look at the chart, if you notice, are we becoming more positive or becoming more negative? We're becoming more positive. Okay? So the cell itself, again, is becoming more positive. And remember, in order for us to reach an action potential, it has to reach the threshold. And the threshold, if you remember, is -55, which is about right here. And if you notice, right at this point is when that action potential happens, and we have this extreme peak of depolarization. It depolarizes, meaning that we're going closer to zero. We actually go a little bit farther than zero, and it starts to do what we call repolarize, which means that we're going back to having a negative charge. And if you notice, remember, our resting membrane potential is -70. This actually goes a little bit lower than -70. So that's what we call hyperpolarization because it has gone a little bit lower than what we want it to be. And eventually, the resting membrane potential is reestablished. Okay? So let's kind of get our brains thinking a little bit. Then we have sodium and potassium, more sodium on the outside, more potassium on the inside. Okay, but more negative here. Actually, potential difference of a negative 70 at rest. In order for us to become this cell to become more positive, think about what has to happen. In order for this to become more positive, what has to happen? Do we have to have, and we're only talking about sodium and potassium at this point, will more sodium come into the cell? Or will potassium leave the cell? In order for this cell to become more positive, because we're at a negative 70, and this has a positive charge, this has a positive charge. So if these positive charges start to leave, will it become more positive or will it become more negative? If these positive charges start to come in, will it become more positive or will it become more negative? So hopefully you're thinking that if we have more sodium start to come in, that's going to make it more positive. And that's pretty much what happens here, an influx of sodium. And that's what's going to come up on these next few slides, okay? So, and this is probably a lot better picture than what I was showing, okay? So at rest, we have this negative 70 resting membrane potential. We have a stimulus or a stimuli, and it's going to help us to reach this negative 55 millivolts. The way that this happens is we're going to have more positivity on the inside of the cell. The way for that to happen, we have to have an influx of these positive sodium ions to come inside the cell. And so as those positive sodium ions start to come inside the cell, it becomes more positive. So it goes from negative 70 to a negative 55. And at that point, we have reached the threshold, which now means that we can have an action potential and it will continue to depolarize, which means more and more sodium will start to go inside the cell until it kind of depolarizes to 0. Okay? Now, we don't want this to continue to happen. So let's go back to this chart. So it depolarizes. This is all the sodium going in, sodium going in, sodium going in, sodium going in. Okay, it's just a little bit too much sodium going in. We're too depolarized. Around this point, which the next slide says is around positive 30, the cell starts to repolarize. And that's this part right here. So if we're trying to repolarize, which means that we want to become more negative, what has to happen? These potassium ions have to now leave the cell because they're positively charged. So as the potassium ions start to leave the cell, it becomes back to being more polarized, more negative, closer to the resting membrane potential of negative 70. But it does it so fast that it actually hyperpolarizes just for a temporary moment about to negative 90. And then that sodium potassium pump will help to return it back to the resting membrane potential of negative 70. So again, I'm going to go back to this chart and talk us through it. Resting membrane potential is a negative 70. Sodium potassium are here. We have more sodium on the outside than we do on the inside, more potassium on the inside. It has a negative 70 charge on the inside of the cell. So we have some type of stimulus. That stimulus is going to reach or get us to reach the threshold stimulus of a negative 55. In order to reach negative 55, we have to be slightly depolarized, which means that sodium ions have started to come in, which means we're becoming more positive. After we reach this threshold stimulus, it's going to cause an action potential to happen. So we have a lot of depolarization, sodium entering, becomes a more positive, until we reach about positive 30. At about positive 30, the cell starts to repolarize. At this point, potassium leaves. You may want to write all this on the chart. It may be even helpful. Potassium leaves. Potassium is leaving. We're becoming more negative. We go dip down just a little bit over, or a little bit farther than our rest membrane potential, about negative 90 or so, a little bit farther than 70. And then we hyperpolarize, and then it reestablishes the rest of the membrane potential. And it says hyperpolarization. It could get to about negative 70. Now, the chart before didn't show going down to negative 90, but after it passes negative 70, that's still considered hyperpolarization. Okay? This is just the same thing, just in word form. So I'm just going to kind of talk through this, just again, kind of reviewing. So after we have again so many sodium and potassium channels along this area, specifically on the axon hillock, what is the trigger zone that's going to cause an action potential to go down an axon. All right. So we have sodium and potassium, they're closed. In order for us to reach the threshold of negative 55, those sodium channels have to open. Okay? Remember, this threshold is negative 55. Right? And so we start at a negative 55. No, I'm sorry, we start with our rest membrane potential at negative 70. And then we work our way to a negative 55. Okay? And we reach that because those sodium starts to come in. It's going to continue to depolarize as sodium diffuses into the cell until they reach about positive 30. And that's going to be our action potential. And then eventually, those sodium ions or channels are going to close up. Potassium is going to open so that we can start to repolarize, so it can go back down towards our rest of membrane potential. But it will slightly hyperpolarize below that negative 70. Sodium channels are going to close because we've hyperpolarized too much. And then the sodium potassium pump is going to help restore this negative 70 rest of membrane potential. Again, this is just kind of showing how those nerve impulses go down an axon. These are the sodium and potassium basically going in and out of the cell, and how the charges kind of change as that nerve impulse travels down the axon. Okay? A thing about neurons is that if it reaches the threshold, it's going to respond or it won't respond. It's going to have a complete action potential or it won't. That's what the all or none response is. If it gets its threshold and it responds, it's going to respond completely all the way down the axon, or it won't respond at all. Okay? And all the impulses are going to be the same strength. And if you have a stimulus that has a greater intensity, which means it's a bit stronger, it's going to cause a higher frequency of those action potentials, but the impulse itself won't be stronger. It'll just have more action potentials that will go down that axon. Now, we don't want a neuron to just be overly active, so it has to have a bit of a rest, and that rest is what we call a refractory period. So we have two parts of the refractory period. One is called the absolute refractory period. Absolute is going to be the time when the threshold stimulus cannot generate another action potential, which means that when we are at that negative 55, we can't generate another one. So just very slightly until that action potential has happened, the channels will be just slightly unresponsive. And so relative refractory period is the time when a high intensity, which we were just talking about, if the stimulus is high enough, it could possibly generate another action potential. Okay, especially if it has not repolarized completely. This is something we've talked about, especially these first few bullets where myelin helps speed it up. It acts as this insulator, very similar to the cords that you have over your maybe phone chargers, computer chargers, a lamp plug, whatever, TV cord, to help to increase the flow of that electricity. Same thing for nerve impulses, and that's why we have to have myelin. We also have those gaps. If y'all remember, the gaps between the myelin sheath is called the node of Ranvier. And I mentioned very briefly that those spaces actually help it to move even faster. And the process of the impulses jumping from one node to the next node is called saltatory conduction. I think there's a picture on the next slide where we'll kind of go over this a little bit, but it actually jumps from one node to the next, and it really helps it go a lot faster. Another thing as far as the impulse conduction, we know that if it is myelinated, it's going to go a lot faster. But also the axon diameter is going to affect it. So it's very thick, it's going to transmit a lot faster than a thin axon. So that's why this is here. If it's thick and myelinated, it's going to transmit so much faster than a thin one that's unmyelinated. Or this one will be, if it was a thick unmyelinated one, this one will still be the fastest type of nerve impulse that will be transmitted. This is the picture of the saltatory conduction that I've been referenced to. So the current will travel in the direction of the axon, but saltatory conduction makes it actually kind of jump from one node to the next. So it's very quick. Instead of, it kind of eliminates the time it takes to go through all of this. It's able to just kind of jump from one node to the next. Makes it very quick. So, a little bit more about synaptic transmission. We know that it's going to be taking information, or nerves can take information from one nerve to another, nerve to a gland, nerve to a muscle. This is specifically talking about from one neuron or nerve impulse to another neuron. And we know that they're going to be responsible for releasing neurotransmitters, and those neurotransmitters have to go through the synaptic cleft, and they then bind to receptors that are on the postsynaptic neuron, which is what we've mentioned before, right? And so the types of neurotransmitters that are going to be released can either excite the postsynaptic neuron or inhibit, okay? And so if it's excitatory, it's going to be able to increase the permeability of sodium, because remember, when sodium starts to come in, that's what causes the cell to reach the threshold and also to have an action potential to become more positive. Or to inhibit it, which means to stop something from happening, um, and that's going to move it away from the threshold, which means the impulse would not happen. Same thing here. It's just synaptic potentials. It says the membrane change in which a neurotransmitter opens the sodium channels. Same thing we were just referring to. This is just specifically talking about the postsynaptic potential. Remember, postsynaptic is after the synapse, so the potential of it to open the sodium channels for excitatory postsynaptic potential. That means that these sodium channels will be open and it could possibly depolarize towards the threshold stimulus and toward the action potential because and then it's going to be, of course, more likely. Inhibitory is going to be just the opposite. Those sodium channels are going to be open, which are going to make the sodium channels leave out, or even chlorine channels. Remember, that's going to make it more negative. And so, if that's going to allow those chlorine channels to come back in, then the inside of the cell will be more negative, possibly even hyperpolarized, especially if those chlorine ions start to leave or start to come in and the potassium ions start to leave. And so, that is not going to reach the threshold, which means that it won't have an action potential that's going to happen because it has inhibited this postsynaptic neuron. Okay? Now we know that neurons don't work independently of each other. A lot of times we have a lot of neurons that are acting on one. Multiple neurons are always just kind of acting on several neurons at a time. And so, depending on if it's an excitatory or inhibitory synapse, this is going to determine what that neuron, that postsynaptic neuron, is going to do. So all of that together, depending on the type of neuron that's being transmitted or the type of signal, or if it's inhibitory neurotransmitters or whatever, that's going to cause this postsynaptic neuron to either be excited or not excited. It just depends. So this effect will depend on the summation of either excitatory synaptic potentials or inhibitory postsynaptic potentials. Okay, and of course, it's going to determine whether or not the signal is transmitted down that trigger zone, which is what we call the axon hillock, which is the very first part of the axon where it will actually start the nerve impulse if it were to happen, if the summation causes it to reach the threshold. And this is several neurotransmitters. You don't have to just memorize every one, but I do want you to know that there are multiple that we have out there. We have the acetylcholine that you may have heard of before many times, especially within the muscular system. But we have that within the CNS, we have the PNS, muscle contraction. We also have things like norepinephrine, dopamine, and they all kind of do different things. I won't ask whether or not certain neurotransmitters are excitatory or inhibitory, but do know that different neurotransmitters can do different things. And they're all going to be carried to the synaptic knob and released via exocytosis. And again, I'm not going to ask what conditions are going to be an effect of different or associated with different neurotransmitter imbalances, but I just want to kind of point out a few. For instance, let's see, insomnia, which most of you have heard of, the inability to sleep, is because we have a deficiency of serotonin. Serotonin is a neurotransmitter, or if we have too much serotonin, we have excessive sleeping. So, and even Parkinson's is related to deficiency of dopamine. Also, drugs are going to alter neurotransmitter levels. For instance, cocaine. It affects dopamine and blocks the reuptake of it. And so you have this euphoria or a sense of euphoria. Nicotine, it affects two, acetylcholine and dopamine, and it can increase alertness and this sense of pleasure. This we mentioned several times already. This is just what happens when a neurotransmitter is about to be released. We know that it's being taken along the axon until it gets to the synaptic knob. We know those calcium ions are going to then infuse or not or be taken into it's going to be taken into the synaptic knob of that presynaptic neuron. At that point, the vesicles will release the neurotransmitters via exocytosis within the synaptic cleft, and those neurotransmitters will then bind to the receptors on the postsynaptic neuron to cause a an impulse or some type of excitatory or inhibitory response. We also have neuropeptides, and neuropeptides are just proteins that are found that can be produced by neurons, and some of them can also be act as neurotransmitters or neuromodulators that can alter the way that a neuron would usually respond to a neurotransmitter or even block the release of a neurotransmitter. Some examples, you may have heard of them, enkephalins. This is what we have naturally to help to block the sense of pain, or also something else that helps to relieve the sense of pain, endorphins. This is what we naturally produce, for instance, during labor, or if you break a bone and it does not hurt immediately, or if you're in a fight and you're getting hit hard, really kind of get your butt whooped. Um, you may not feel the pain in the moment. Now, after it's all said and done and those endorphins have worn off, you will feel the pain. But we kind of naturally secrete endorphins to do that. And also, substance P is just another type of neuromodulator that helps to kind of conduct those pain impulses as well. And this is actually going to, or enkephalins and endorphins, the first two we just talked about, actually inhibit the release of substance P. One thing that's kind of interesting about the man-made opiate drugs like morphine or heroin and opium, um, we all know that these are painkillers, and of course, we all know that they are abused, right? And but the thing is, we produce natural opiates, and those opiates are the endorphins that I was just referring to. And so, what happens when people take opiates for pain, which a lot of times is perfectly fine because that's what doctors prescribe, but what happens when we abuse them is that our body, and that's what this very last bullet is referring to, our body um, is becomes accustomed to these type of opiates to do the job that our natural endorphins are supposed to do. Which means when we try to get off of these opiate drugs, our natural endorphins that we're produced are no longer being produced because our body has become dependent upon endorphins. And so now that we're dependent on these, when we try to get off, we experience pain and withdrawal symptoms. Okay, and it's because our body is not producing those own endorphins that we're supposed to because it has become dependent on these. It's kind of interesting. The last little section is about impulse processing and just a few slides, kind of how they kind of work together. So we also have neural pools. Again, remember I said that neurons do not just work independently of each other. We have groups of neurons, specifically interneurons, they're going to connect with each other in the CNS, also different parts of the CNS to get the job done. Okay, and they're going to be receiving information from other neurons and also taking information out to possibly other neural pools or to muscles or glands. Okay. Um, and of course, it has all to do with those neurons that are taking in or releasing or taking in information, or the bringing in information to these neural pools. So it has two ways that they can kind of work. The first is convergence, which basically means that we have two neurons within a neural pool. They're acting on another. And again, depending on if they're excitatory or inhibitory, it will cause this neuron, the postsynaptic neuron, to do what it needs to do. And then the last one, divergence, we have this one neuron that the collateral kind of splits and branches off into two. And depending on if it's excitatory or inhibitory, will cause it to process it differently. And that is the end of this part of the nervous system.